the aggressiveness of the male towards the female. Now we will be talking about the biology of heredity, and under that we'll be talking about genetics. Genetics is a biological science of heredity which attempts to explain both the similarity and the differences between parents and offspring. So genetics has to deal with parents and offspring, considering the similarities and the differences between them. The study of that is called genetics. Heredity is a passing on or transmitting of traits or characters from parents to offspring. So you cannot talk about genetics without talking about heredity, talking about genes. So when characters, the passing on of characters or traits from parents to offspring or to their children is what we call heredity. And examples of heritable characters include blood groups, rhesus factor, and the rest. Color of skin is even heritable because most of those things are passed down from parents, and that is why you look like your parents. And somebody has not even seen you before and has seen your dad and say, "Oh, you are so person's daughter." So it's something like that. So heredity is the passing on of or transmitting of traits or characters from parents to their offspring. Now we're talking about the monohybrid inheritance. In the course of this class, we'll be looking at the Mendel's laws, the Mendel laws, first and second law, majorly. Monohybrid inheritance is the inheritance of a single characteristic, and it follows the Mendel's first law. And the Mendel first law is also called the principle of segregation. The characteristics of an organism are determined by internal factors which occur in pairs, and that is called alleles. Mendel dealt with one characteristic and crossed a pure tall plant with a dwarf plant and found that all the seeds resulting from the cross gave rise to a tall plant. A tall plant and a dwarf plant, he crossed them and discovered that all the offspring resulting from that cross are all tall. And the offspring he called the first filial generation or the F1 generation. So then after that, he self-pollinated those F1, that is, he crossed the offspring now. And the result, that those ones gave rise to a set of offspring which he calls the second filial generation or the F2 plants. And he noticed that in this offspring of the second filial generation, that they were either tall or dwarf in the proportion of ratio 3:1. That is the ratio of dominant to receive phenotype. The tallness that overshadowed dwarfness in the first generation is said to be dominant, while the dwarfness is said to be recessive. These conflicting pairs of characters are called allelomorphic pairs. So the Mendelian experiment is what I want to illustrate for you. I said you crossed a tall plant with a dwarf plant. So let the tall plant be represented by TT. So this is the tall plant, crossing it with a short or a dwarf plant. Dwarf. Then this one is a tall plant. And in this law of principle of segregation, then the gametes from these are the parents' plants now, parents. Then the gametes, we have a gamete. Since they are the same, one came out, then another came out. So by the time you cross them, you have a tall plant, even though this resultants or these results is a combination of the tall and dwarf when you look at the gene, because this is represented as the gene, the genotype. But when you look at the phenotype, that is the physical appearance, the phenotype is the physical appearance of a particular plant or animal, while the phenotype is the genetic makeup. So the physical appearance of these plants is tall. They are tall. They are tall, even though it is not like this tall parent, but all of them. By the time you cross this tall plant and this dwarf plant, you discover that all of them, the first generation plant, that's the first filial generation, F1 plant, they are all tall. Now, by the time he crossed these ones, since they are all tall, TT, all tall. Now he now did a self-crossing or self-pollination of this plant now, bringing two of them together. Okay, so this is the second generation, the resultant. This is F1 plant. After the crossing of the tall plant and the dwarf plant, this is the resultant. So he did a self-pollination for them. So by the time you do a crossing, let's do it together. This TT, so you have TT, then this one with this, you have TT, then you have this with this, you have this again, then this with this, you have. So looking at this, you can see that this is a dwarf plant coming back now. This is dwarf. This is tall. This is tall. This is also tall. So that is why we said that it came about both after when you got the second generation or second filial generation, that's the F2 plant. The proportion of tallness to dwarfness is 3:1. 3:1. So that was the experiments that Mendel discovered. But another thing is that now he said this tallness, this capital T, are the dominant factors or the dominant genes, while these ones are the recessive gene, and that is why this one is a tall plant. Even though it has this, it is not the plant is not expressing this dwarfness. You know, this is an element of dwarfness, but by the time it was 2 here, that was why it manifested the dwarfness. So capital T, tallness is dominant over dwarfness. The tallness overshadowed the dwarfness in the first generation, and that was why there was no dwarf plant in the when you cross the tall and the dwarf plant, there was no dwarf plant. So the tallness was said to be dominant, while the dwarfness is said to be recessive, and these two are called allelomorphic pairs. They are allelomorphic pairs, so they are called allelomorphic pairs. The Mendel first law is the law of segregation of genes, and it states that characters of an organism are controlled by genes occurring in pair. Genes occurring in pair. So you see they are in pairs, not just one. Then they are independently transmitted in gametes during meiosis from one generation without undergoing any alteration. I take that again. The Mendel's first law, or the law of segregation of genes, states that characters of an organism are controlled by genes occurring in pairs. They are independently transmitted in gametes during meiosis from one generation without undergoing any alteration. Now, a pair of allelomorphs or alleles are separated or segregated when gametes are formed. That was when they are separated, they will come individually, since each is present on the homologous chromosome. So they separate or they are segregated when the gametes are formed. Now, that is for monohybrid inheritance. Now we move on to talk about the dihybrid inheritance. Dihybrid inheritance involves 2 pairs of alleles and forms the basis of the Mendel's second law, also known as the principle of independent assortment. That is, any pair of characteristics may combine with either or another pair. The resulting phenotype is approximately a ratio of 9:3:1. You know, this one we had 3:1. That's the ratio of in monohybrid inheritance, ratio 3:1. But in dihybrid inheritance, it is 9:3:1. In the monohybrid inheritance, it was in the example that I illustrated, it was only the height that was considered. But in this dihybrid inheritance, we will be discovering that it's not only the height. So this is TTR capital. So it is representing tall, round seeds. They are tall and they are round. We now want to cross it with a dwarf wrinkled seed. Dwarf wrinkled seed. So just like this. Now, so the gametes now that will come from this will just be T, R, then this will be T, R2, the short plant. Then for the first generation, F1, let's do it together. Then this one, this, so we have T, T. For the dihybrid inheritance, I said we want to cross a tall round seed with a dwarf wrinkled seed. Dwarf wrinkled seed, tall round seed. So from that we have these gametes. TR, the capital TR, then we have capital TR. By the time we do a crossing for the first generation, we have this one. This is the first generation, TTRR. Like we did in the Monohybrid Inheritance too. When we now do a self-pollination for this one, TTRR, these are the gametes that we came about, and there are 8. But because of the ambiguity, we can't do the straight crossing, just as I did for the monohybrid inheritance, and that's why we bring it to this square, or this box-like structure called the Punnett square. Punnett. So the Punnett square is what will help us in solving the dihybrid inheritance. So these 8 gametes, TR, the dominant T and R, the dominant T, recessive R, recessive T, dominant R, recessive T and R, dominant T and R, dominant T, recessive R, recessive T, dominant R, then dominant T and R. So that's why we brought it here. Since they are 8, we put 1 here, we put the other 4 here. So TT, TRTR, you have TTRR, then